You know, when you swallow a pill, you are essentially like flying blind.
Oh, completely.
Right. You take it, it dissolves in your stomach, and then you just have to hope that those invisible chemical compounds are somehow navigating the dark microscopic soup of your body to actually do their job.
Yeah, you just swallow and wait.
Exactly. The actual mechanics of it are completely hidden from us. We rely on, complex blood tests or even invasive biopsies days or weeks later just to tell us if that invisible chemistry actually worked.
It is a fundamentally abstract process, really. I mean, for decades, the gold standard in medicine has basically been waiting for systemic chemical proof that a therapy is effective. You can't just look at a patient and immediately know a drug has halted some microscopic cellular process.
But what if you could? Okay, let's unpack this. What if you didn't have to guess? Because today we have a really fascinating mission.
We do.
What if you could literally take a photograph of your cell, feed it to an artificial intelligence, and watch the exact physical moment a drug alters your body's chemistry? Today, we are diving into a groundbreaking paper from the journal Cell, detailing a new machine learning framework called deep phase.
It's a fantastic paper.
It really is. And our mission for this deep dive is to explore how this AI is analyzing shifting microscopic blobs inside our cells, and how those shifting shapes might completely revolutionize the speed of drug discovery. So for everyone listening, if you've ever taken a medication, this completely flips how we understand what that drug is doing inside you.
The premise here is a massive paradigm shift. I mean, deep phase essentially argues that the physical architecture of our cells is a direct, real-time reflection of their chemical health.
A physical reflection of chemical health. That's a great way to put it.
Right. If we can understand how to read that architecture, we can decode exactly how drugs are functioning, like instantly.
But to understand how the AI reads the architecture, we really have to understand the weird, shape-shifting biology it's actually looking at. Because inside our cells, there are these structures called biomolecular condensates. And they are completely unlike the normal organs we picture when we think of biology.
Right, yeah. Most of us learned in high school biology that the parts of a cell, like the nucleus or the mitochondria, they're organelles wrapped in a protective membrane.
Right, the classic textbook diagram.
Exactly. You can picture them as little water balloons floating in the cellular fluid. But biomolecular condensates, however, are entirely membraneless. They have no skin, no protective barrier at all.
Wait, so how do they even exist if they don't have a skin holding them together? Why don't they just, dissolve into the rest of the cell?
Well, it comes down to a really fascinating physics concept called liquid phase separation, or LOPS, if you want the acronym.
Liquid phase separation.
Yeah.
Okay.
Yeah. So certain proteins and RNA molecules inside the cell have specific physical properties. They have these intrinsically disordered regions, sticky regions, basically, that make them want to huddle together.
So it's literally like dropping olive oil into a bowl of water.
Yes.
The oil doesn't have a membrane around it, but the droplets still pool together into these like floating blobs just because of their physical properties.
That is a brilliant way to visualize it. These molecules condense into liquid droplets right there in the cellular fluid. They act as dynamic, highly concentrated hubs for complex cellular functions, and the largest, most prominent of these oil drops inside the nucleus of your cells is called the nucleolus.
The nucleolus. And what's wild to me about the nucleolus is that it's not just one uniform drop of oil.
Oh, not at all.
It has a highly specific, painstakingly detailed geometry. It is composed of 3 distinct concentric phases.
Yeah.
I like to picture it like a layered gobstopper candy or, one of those nested Russian dolls. You have an inner core, a middle layer, and an outer shell.
That's right.
And each layer acts as a different department in the cell's ribosome factory.
Let's actually break those 3 factory departments down, because understanding what happens in each layer is essential to understanding the AI's eventual discovery later on.
Yeah, let's do it.
So the innermost core of this Russian doll is the fibrillar center, or FC. Let's call this the factory floor. This is the space where raw ribosomal RNA is first transcribed from your DNA. It is marked by specific proteins like RPA16. It's the absolute start of the manufacturing process.
Okay, so the FC is the factory floor where the raw materials are made. Then you step outward into the middle layer.
Right, the dense fibrillar component, or DFC.
DFC, got it.
If the core is the factory floor, this middle layer is the assembly line. This is where that newly transcribed raw RNA gets processed, folded, and chopped up into exactly the right pieces. And this layer is marked by a different protein called NOP-56.
Okay, so factory floor in the middle, then assembly line around that.
Precisely. And once the pieces are processed on the assembly line, they migrate outward to the final shell, the granular component, or GC.
The GC layer.
Yes, this is your packaging and shipping department, marked by the protein NPM1. It's where the large ribosomal subunits are fully assembled and prepared to be shipped out to the rest of the cell.
Okay, here's what I need to clarify, though.
Yeah.
You've got this layered fluid factory. What happens if the chemistry stops?
That's the million-dollar question.
Right, like if I take a drug that shuts down the conveyor belt on the assembly line, does the physical building itself change shape? Does the fluid factory literally collapse?
It absolutely does, and this is the core concept. The physical structure of the nucleolus is actively maintained by the continuous flow of chemical reactions happening inside it. If you use drug to halt transcription on the factory floor, or a different drug to block processing on the assembly line, those concentric liquid layers physically deform in wildly different ways.
So if I'm tracking with you, the actual geometry of our cellular hubs isn't static. It's constantly morphing based on the real-time chemistry going on inside.
Exactly.
Which makes you wonder, if these tiny liquid factories are always reacting and shifting, how much is the internal shape of our bodies changing right now without us having any idea?
It is a profound realization.
Okay, before we move to how the AI actually sees all this, let's do a quick bullet recap for everyone listening.
That idea.
Because that was a lot of deep biology.
It was.
So bullet 1, biomolecular condensates are membraneless cell hubs like oil drops in water.
Yeah.
Bullet 2, the nucleolus is the biggest one.
Yeah.
And has 3 functional layers. The FC factory floor, the DFC assembly line, and the GC packaging department, all dedicated to making ribosomes. And bullet 3, these layers physically change shape if the chemistry inside them is disrupted.
Spot on. That is the perfect summary.
Awesome. And for you listening, I just want to ask, have you ever considered that the physical microscopic geometry of your cells is actively shifting based on the chemical reactions happening inside them? It's just wild.
It really is. And for decades, scientists actually knew this was happening.
Yeah.
They knew that when you disrupted the chemistry, these condensates changed shape. But here is the massive roadblock they faced. How do you accurately measure a shifting, membraneless fluid blob without human bias?
Right. Because if I look at a cloud, I might see a dog. And you might look at the exact same cloud and see a car.
Exactly.
If scientists are manually deciding what a deformed blob looks like, they are just injecting their own subjective assumptions into the data.
That is precisely the problem. In the past, scientists relied on traditional image analysis software to try and automate this, like Cell Profiler, for example. They would tell a program to look for specific, handcrafted features. A human would decide, I think size is important, or I think circularity matters, and code that in, but that still limits you to what the human thinks is relevant. It completely misses the hidden patterns.
And this is where the computer science kicks in and solves the bias problem, right? The researchers built DeepPhase.
They did.
They used a specific type of AI architecture called a ResNet-50 convolutional neural network, or CNN. And the magic of a CNN, from what I understand, is that you don't tell it what to look for at all.
Not at all. It learns directly from the raw pixel data. The researchers took thousands of high-resolution fluorescent microscopy images of human U2S bone cancer cells.
Bone cancer cells.
Right. They fed those raw pixels into DeepPhase, and the AI autonomously learned to map the cells into what we call a two-dimensional activation space.
Okay, let me visualize this activation space. It's basically a massive digital map, right?
Yes.
DeepPhase takes every single cell it looks at and drops it onto this map as a coordinate point based purely on its shape. So to train it, they first showed the AI completely normal, healthy cells, and those cells all clustered together in the center of the map.
Correct. That establishes the baseline, the normal phenotype. Then, the researchers treated a new batch of cells with a specific drug, a transcription inhibitor called CX-5461.
CX-5461.
This drug shuts down the inner core, the factory floor. When that happens, the nucleolus deforms into a shape the researchers called the cap morphology.
The cap, like a hat.
Sort of. The inner and middle layers literally migrate to the outer edges of the structure, looking like little fluid caps sitting on top of a larger sphere.
Oh, I see. And DeepPhase plots all those cap cells in a completely different region on its digital map, say way up in the north corner.
Exactly. Then they used a second drug, a processing inhibitor called flavopyridol, or FVP.
FVP. And this one breaks the middle assembly line, right?
Right. And that caused an entirely different shape called the necklace morphology. The outermost packaging layer, the GC, it fragments and breaks away, looking like scattered beads on a necklace. And the AI plots all of those cells way down in the south corner of the map.
So it visually separates the different chemical failures. That's amazing.
But what is truly revolutionary here isn't just that the AI can sort a cap from a necklace. What's fascinating here is what happens when the researchers feed varying doses of these drugs into the system.
Oh, like ramping up the dosage.
Yes. As you slowly increase the amount of the drug, the AI tracks the gradual microscopic transition of the cells traveling across that digital map. From this journey, the AI calculates a morphological response curve. It generates a value called the RC50.
The response concentration 50, I want to make sure I have this right. This is the exact amount of the drug required to cause a 50% change in the physical shape of the cell.
Yes, exactly. And here is where the paper delivers a true breakthrough. They compared the AI's visual RC50 value to the drug's biochemical IC50 value.
Okay, let's make sure we define that. The IC50.
Yes.
The inhibitory concentration. That is the traditional hard chemistry measurement of how potent a drug is, right?
Correct.
It's the exact concentration needed to kill half of the target chemical activity in the cell.
Precisely. To find an IC50, human scientists have to run incredibly complex, expensive, and time-consuming chemical assays. But when they compared the numbers, the visual RC50 generated by the AI perfectly matched the biochemical IC50.
Wait. Hold on, let me make sure I'm really grasping the weight of this.
It's a big deal.
Are you saying this AI skips all the tedious chemistry entirely? It just looks at the raw pixels of a deformed cell, tracks its shape on a map, and perfectly predicts the chemical math of the drug's potency. That must cut out, months of lab work.
It bypasses an enormous bottleneck in drug discovery. You are suddenly deriving highly complex quantitative biochemical data purely from a photograph. The physical disruption perfectly predicts the chemical potency.
Okay, time for another quick bullet recap, because my mind is a little blown.
Let's do it.
Bullet 1. Deep phase AI uses raw images to categorize nucleolar shapes without human bias, plotting them on a map.
Yeah.
Bullet 2. Different drugs cause different shapes, like the cap or the necklace. And bullet 3, the big one. The AI accurately predicts a drug's biochemical potency, the IC50, purely from tracking morphological changes, the RC50.
Perfect recap.
So for you listening, just think about this. If an AI can predict a drug's exact potency just by looking at a photograph of a cell, what does this mean for the future speed of drug discovery? It's incredible.
It uses the entire timeline.
But, any good scientist is going to push back and ask, is this just a nucleolus expert?
Oh, absolutely.
Right. Does this AI only know how to look at this one specific Russian doll structure? Or is it a universal tool?
That was the immediate hurdle the researchers had to clear to prove the platform was actually robust. They had to take deep phase outside the nucleolus and point it at completely different biological environments.
Okay.
So first, they looked at nuclear speckles.
Right. Nuclear speckles. I know these are entirely different membraneless condensates. Their job isn't making ribosomes, right? They act as hubs for splicing messenger RNA.
Exactly. And just like the nucleolus, they change shape when their specific chemistry is disrupted. The researchers treated cells with flavopiridol, that drug that breaks the assembly line, and the AI detected that the speckles morphed into a round shape.
Just totally round.
Yes. Then they used a completely different drug called pladanolide B, or PLAD-B. This one directly attacks the splicing process, and the AI mapped the speckles becoming dilated.
Here's where it gets really interesting, though, because I was reading the source, and they didn't even limit this test to human cellular structures.
No, they didn't.
They brought in a virus. They infected cells with respiratory syncytial virus, or RSV. Now, when a virus like RSV infects you, it hacks your cellular fluid. It forces your cell to build foreign viral condensates, known as inclusion bodies, so the virus can replicate its own RNA.
It basically builds its own rogue factories inside.
Rogue factories, yeah.
And when the researchers treated these infected cells with an antiviral drug called cyclopamine, those rogue viral factories changed from spherical to aspherical. And yet again, the AI tracked the changing pixels, calculated the visual RC50, and it perfectly matched the antiviral drug's functional potency.
And this is where they pitted the AI against the older traditional software, right? The cell profiler programs where humans handcrafted the features. How did the old software do with the virus?
It failed completely.
Wow.
Viral infections create a lot of visual noise. The cell is under attack, things are messy. The traditional software couldn't reliably separate the viral blobs from the background chaos. It couldn't handle the noise because it was only looking for the rigid, specific features it was programmed to find.
But the AI crushed it. Because it was looking at raw pixels, it didn't care about the noise, it just found the underlying mathematical pattern. Okay, quick recap on this section. Bullet 1. Deep phase successfully analyzes other human structures like nuclear speckles. Bullet 2, it can even analyze foreign viral inclusion bodies like RSV. And bullet 3, it proves to be a robust, universal platform that is way superior to older, future-based image analysis.
It really is acting as a universal translator for the cell.
Yeah. So for everyone listening, what other mysterious shape-shifting structures in biology could this AI unlock next? It looked at human speckles, it looked at viral invaders, and it just didn't blink.
And that exact curiosity is what drove the final and perhaps most exciting phase of this research. The team knew the tool was bulletproof. It worked on known biology. So they decided to unleash deep phase into the complete unknown.
The unknown.
Yes, they wanted to see if it could discover something human eyes had entirely missed.
So they set up a massive blind test. They took a library of small RNA-binding molecules, so basically a vault of potential untested drugs, and dropped them onto thousands of cells. Then they just let deep phase analyze the images to see what happened.
A huge screening process.
Right. And for the most part, the AI categorized the reactions as expected. Most of the drugs just created those familiar cap shapes. But then it hit a drug called topotecan, or TPT.
TPT.
And the AI basically threw its digital hands up in the air. It gave topotecan a no-call classification.
Now, in the world of machine learning, a no-call might sound like a failure, but here it was a massive success.
Oh, really? Why?
Because it means the AI confidently determined that the shape it was seeing was not normal, it was not a cap, and it was not a necklace. It was something entirely unrecognized by the map. It knew what it didn't know.
And because the AI flagged this anomaly, the human researchers went in and looked at the images themselves. And they found a brand new, previously undiscovered nucleolar structure. They named it the flower morphology.
The flower. It is an incredibly unique, inverted structure. Think back to the necklace shape we discussed earlier, where the outermost packaging layer fragments and completely separates from the middle assembly line.
Right, the scattered beads.
Exactly. But in the flower, that outer granular component layer actually stays in contact with the middle layer. It wraps around it, clinging to it like petals on a flower. The boundary stays intact. The question was, why?
Right. To understand the why, we have to look at what that specific drug, topotecan, actually does to the cell. Topotecan is an inhibitor that targets an enzyme called TOP1.
TOP1.
And TOP was a DNA to polymerase. Okay,
Yeah.
Let's break that down for everyone. When DNA is being unpackaged and read by the cell, the physical strands get violently twisted and supercoiled. If you ever tried to pull apart a twisted phone cord or like a tangled skein of yarn, you know how the tension builds up ahead of the pull until it knots completely.
That tension is fatal to cellular processes. So TOP1 acts as a pair of molecular scissors. Its entire job is to make tiny temporary snips in the DNA strand to relieve that twisting tension, unspin it, and reconnect it so the reading process can continue smoothly.
So topotecan stops the scissors. It stops TOP1 from relieving the tension, so the DNA just gets hopelessly tangled.
Yes, it stalls the whole thing.
But I'm still trying to connect the dots here. Why does tangled DNA create an intact flower shape, while the other drug we talked about creates a broken necklace shape?
If we connect this to the bigger picture, the mechanism boils down to cellular trash.
Crash.
Yes. Remember the drug that causes the necklace shape, flavopiridol? It breaks the middle assembly line, but it leaves the inner factory floor running. The core keeps transcribing raw RNA, pushing it out to an assembly line that is completely broken.
Wait, let me visualize this. If the assembly line is stopped, but the factory floor keeps blindly making raw parts,
Yeah.
those parts are just going to pile up on the conveyor belt.
Exactly.
There's nowhere for them to go. You get a massive buildup of misprocessed RNA junk.
Exactly. The junk physically piles up. And because these condensates are just liquid layers held together by delicate physical properties, that massive buildup of debris physically bursts the boundary between the middle layer and the outer layer. The connection breaks, creating the scattered beads of the necklace.
So it is fundamentally a structural integrity issue. It's like a dam bursting because way too much debris and water built up against it. The physical walls just give out.
That is the perfect analogy. Now apply that to topotecan, which creates the flower. With topotecan, there is no junk buildup. Because TOP1 is inhibited, the DNA gets so tangled so early in the process that the inner factory floor can't even produce those raw RNA parts.
Oh, I get it. Because there is no debris flowing down the river, the dam holds. The boundary between the middle layer and the outer layer stays perfectly intact, wrapping around like flower petals.
Precisely. And the researchers didn't just hypothesize this. They proved the dam analogy in the lab with a brilliant experiment.
What did they do?
They took a batch of cells and treated them with the assembly line breaker first, phlegopyridal. So the factory floor pumps out parts, the junk builds up, the dam bursts, and you get the disconnected necklace shape.
Okay, the river is full of debris.
Then they took those exact same cells and washed them with the transcription blocker drug, CX-5461. They actively shut down the inner factory floor.
They stopped the flow of debris at the source.
Exactly. They halted the transcription, which allowed the cell to clear out the existing junk buildup. And the absolute moment the junk was gone, the broken nucleolus morphed right back together into the connected flower shape.
That is incredible. They proved definitively that the microscopic physical boundaries inside our cells are actively maintained by a breathtakingly delicate balance of chemical production and chemical processing.
It is a stunning biological discovery about how enzymes act as actual structural architects. And it is a perfect example of human-AI collaboration. The AI didn't solve the mechanism on its own. It couldn't tell us why the flower existed, but it confidently flagged the anomaly on its digital map so that human intuition and experimentation could step in and decode it.
Okay, final bullet recap before we close this out. Bullet 1. Deep phase identified a novel flower morphology caused by TOP1 inhibition. Bullet 2. The flower happens because there's no junk buildup, unlike the necklace shape. And bullet 3. This revealed that nucleolar structural boundaries are maintained by the exact balance of RNA transcription and processing.
A beautiful summary of a really complex mechanism.
Thanks. So for everyone listening, how amazing is it that an AI's error, a no-call, actually led human scientists to a brand new biological discovery about how enzymes act as structural architects.
It's the future of science, really.
So synthesizing everything we've uncovered today, an AI looked at raw pixels, mapped shifting liquid blobs, predicted complex drug math, and then guided humans to a brand new biological mechanism.
So what does this all mean for you listening to this deep dive? Why should you care about the shape of a nucleolus in a petri dish?
Well, it's because the physical structure of these condensates is intimately, inextricably linked to human health.
Right.
That is the ultimate takeaway. Diseases like cancer, neurodegenerative conditions like ALS, and severe viral infections all fundamentally involve cellular condensates that are broken, mutating, or hijacked. The architecture is sick. And deep phase gives us a high-speed, entirely unbiased map to fix it. We don't have to wait months for chemical assays to tell us if an experimental Alzheimer's or cancer drug is working. We can drop thousands of potential compounds onto disease cells and instantly see, literally see with our own eyes if they snap the broken architecture of our biology back into its proper shape.
It completely removes the manual guesswork from early-stage drug discovery. We are moving from a paradigm of assuming to a paradigm of observing.
We can finally watch the invisible chemistry working in light. Which leaves us with one final, truly wild thought to mull over. If our cellular structures act as these highly sensitive, real-time physical readouts of our underlying chemical health, could we eventually use a simple cellular photograph as a universal diagnostic tool?
Oh, that's a huge thought.
Right. Imagine a future where a quick image scan of your cells provides a visual fingerprint, an instant map capable of detecting the shifting microscopic shapes of a disease in your body, long before you ever feel a single symptom.
It entirely changes how we might view the landscape of diagnosis and preventative medicine in the future.
It really does. It's a whole new way of seeing ourselves. Thanks for taking this deep dive with us.